My name is Charles Manning, and I am a professor at MD Anderson Cancer Center in Houston, Texas. I'm the research lead at MD Anderson's theranostics program and the director of our cyclotron radiochemistry facility. My background is in chemistry, radiochemistry, and diagnostic imaging, and our research programs are focused on developing and translating new therapies and imaging agents that are based on molecules known as radiopharmaceuticals. Radiopharmaceuticals allow us to detect tumors non-invasively and characterize them, giving us a better understanding of their unique features. Subsequently, through a minor modification of the imaging agent, we can use a nearly identical molecule to target the tumor with therapy. In this way, physicians can use imaging to select a therapy that's well suited to treat a patient's tumor, and then even follow the therapy directly and non-invasively to confirm that the drug has reached its target, and importantly, we know how much drug got there. We call this combined approach theranostics, which is a blending of the words therapy and diagnostics, and it represents a unique modality in the armamentarium of cancer medicine. Despite early and impactful clinical success of theranostics in patients with certain tumors, such as neuroendocrine tumors and prostate cancer, we are motivated to deliver this approach to patients with colorectal cancer who do not yet benefit. Our project is based in a strong partnership with key leaders at both ends of drug development at MD Anderson, which is required to reach this goal. And it includes Dr. Scott Kopetz, a medical oncologist and international leader in colorectal cancer research, and Dr. Sam Hanash, an expert in target discovery and proteomics. Collectively, our collaborative team's project, known as radionuclide theranostic for BRAF mutated colorectal cancer, is based in new therapeutics discovery and seeks the first theranostic pair for imaging and therapy, specifically for patients with colorectal cancer. We are excited about the opportunity to work together to translate the breakthroughs of this project directly to patients in need. Overall, theranostics represents an important tool at the physician's disposal that allows detection and visualization of cancer. But to our thinking, most critically, provides highly precise and potent therapy to treat patients and eliminate tumors. Our project's research will deliver a new understanding of how to target colorectal cancer with theranostics specifically, and beyond the specific goals of the present work, will lead to new opportunities to expand an even larger portfolio of theranostics for patients with colorectal cancer. Given the complexity of colorectal cancer, our vision is that a one-size-fits-all approach will not lead to the cures we desire, but rather we need many theranostic approaches to personalize therapy for patients who experience colorectal cancer. The impacts of partnering with the Colorectal Cancer Alliance are not limited to the funding. As scientists and clinicians, we're inspired by the strong presence and perspectives of patients and advocates that join us at the bench and bedside through this journey. This creates a unique and highly motivating ecosystem for discovery. Yet the funding is also vital. There's simply not sufficient funding to rapidly move discoveries in the lab into impactful clinical trials for our patients. Furthermore, funding through the Colorectal Cancer Alliance impacts the types of questions that we can pursue. Rather than focusing on the next incremental advance, the Colorectal Cancer Alliance allows us to think big about groundbreaking opportunities that live in the high-risk, high-reward space. Supporting this type of research requires shared vision, close partnership, and trust, and that makes the Colorectal Cancer Alliance very special.